Hello, everyone, and welcome to today's webinar, The Evolution of Carbon Fixation, Studying Form I Rubisco, using Ancestral Sequence Reconstruction and Mass Photometry. I am Megan Pascal of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Refine. To learn more, visit refine.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well, if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Luca Schulz, doctoral researcher, Max Planck Institute for Terrestrial Microbiology. Luca, you may now begin your presentation.
Hello, everybody, and thanks for the nice introduction. I hope you can hear me just fine. I'm here today to talk about our recent study about the evolution of increased complexity and specificity to dawn of Form I Rubiscos, and I'll dive right into it. Basically, the main question that we set out to answer in the very beginning when I started my research was not so much focused on Rubisco itself, but rather about the evolution of complexity in proteins in general, specifically about the evolution of obligate heterooligomers. And here we were mostly interested or intrigued by the observation that many proteins assemble into obligate heterooligomers. So proteins that are composed of different distinct subunits encoded by distinct genes, such as in a very simple case, for example, Rubisco, which is composed of two distinct subunits, one large and one small subunit, which I'll come to in more detail in a bit. But there's also very extreme examples of this, such as, for example, ATP synthase that contains 17 different subunits that all have to come together to form this very intricate molecular machine. And oftentimes, these obligate heterooligomers actually evolved from less complex ancestors that actually didn't contain as many subunits as these molecular machines contain in present-day organisms. Basically, for any obligate heterooligomere that we observe in nature, there's very often the case where in bacterial genomes or in just related genomes, we can find ancestral forms of the same protein complex that is a bit less complex. And then over a certain interval of evolution, a new subunit was gained. But then also, presumably over the same interval of evolution, this new interaction eventually became essential for the complex. And oftentimes this actually happens without a real change in the function of the complex. It might become slightly better, but this is often not really visible or not easily decipherable. So it actually becomes really difficult to decipher the function of this novel subunit. And a further reason as to why this is actually pretty difficult for these innocuous small interaction partners that are strictly essential for an oligomer is that if you now take this essential subunit away, your protein complex of choice would be insoluble or non-functional. And with this, we pose two questions. The first is just why do such new interactions evolve, especially if there's not really any obvious function associated with these novel subunits? Is there a good way to decipher the function of such a subunit? And why do proteins suddenly start to depend on a subunit that they newly recruited into a complex? You can imagine that this could just be an interaction that doesn't necessarily have to become essential. And for this, we decided to retrace the evolution of Rubisco as our model obligate heterocomplex in the lab to try and answer these questions. Just a very brief introduction on Rubisco itself. Rubisco stands for ribulose 1,5-bisphosphate. So this is this substrate here. This is a sugar substrate that is diphosphorylated. 1,5-bisphosphate carboxylase oxygenase. And its main function in nature is that it carboxylates ribulose 1,5-bisphosphate to produce two molecules of 3-phosphoglycerate. And as such, this reaction is the core reaction of the Calvin cycle, which is used by, for example, autotrophic organisms to fix carbon dioxide. And prominent members of autotrophic organisms that use the Calvin cycle are, for example, plants and algae that are all around us and use Rubisco to obtain its biomass to fix carbon dioxide. In these organisms, Rubisco forms this very typical L8S8. So this is 8 large and 8 small subunits complex that I've already alluded to earlier. which for the sake of my presentation, I will mostly be representing in this schematic, where the large subunits are these sort of bean-shaped subunits that form a ring. And then there's 4 small subunits sitting on top of this octamer of large subunits and 4 small subunits sitting at the bottom of this ring of large subunits. And the small subunits sort of serve to cap this complex. In Rubisco, the small subunit is non-catalytic, so there's also no real obvious function associated with it, but it is strictly essential. And I should mention, it's the reason why there's no obvious function associated with it is, first of all, because it is essential and you can't easily take it away and decipher its function by simple experiments in the lab. But then also because the small subunit is actually not in direct contact with the catalytic center of Rubisco at all. In fact, it's about 20 to 25 angstroms away from it. And also, with this, and this makes Rubisco a good model system for our study here, the small subunit of Rubisco is known to be an evolutionary novelty. So if we look into the genomes of organisms which we know acquired Rubisco earlier than plants or algae, we can see that over the course of evolution, Rubisco transitioned from being a simple dimer of large subunits to eventually becoming an octamer of large subunits that then eventually incorporated the small subunit and formed this canonical, or this very typical L8S8 complex, which in total contains 16 subunits. As I've already mentioned, the small subunit's role is not really known, yet there were some guesses as to what it might be involved in. And this comes from the observation that these L8S8 Rubiscos are mostly found in organisms that occupy aerobic habitats, whereas all the Rubiscos that are in organisms, or all the Rubiscos that don't form a complex with the small subunit, are typically found in anaerobic organisms. And now, why this might be important, or what the small subunit might be involved in, for this it's important to know that Rubisco can actually catalyze a second reaction in which it doesn't take carbon dioxide as its substrate, but instead it mistakenly reacts with oxygen and incorporates oxygen. This leads to a process called photorespiration, in which the 2-phosphoglycolate, which is produced from the oxygenation reaction, has to be metabolized or detoxified in a reaction that releases carbon dioxide and ATP. And now people have presumed that the small subunit might be involved in modulating Rubisco's so-called specificity factor, which is a measure of how specific a Rubisco is for its gaseous substrates and how good it is at discriminating CO2 from O2. However, the sole evidence for this is basically the fact that the L8S8 Rubiscos are mostly found in aerobic habitats where oxygen is present, and the Rubisco complexes that incorporate a small subunit are typically more specific, whereas those complexes that don't contain a small subunit are mostly found in anaerobic environments where there's no oxygen present. All right. Now, with this, we set out to answer essentially the questions that I posed earlier. Why did the small subunit evolve, and what is its function? And why is it now essential when it wasn't in the past? For this, and I'm now going to break this down in 4 further questions that I'm going to be answering in sort of the next few slides. We first wanted to basically find the evolutionary interval over which the small subunit evolved, so that we can then retrace its evolution. For this, we built a phylogeny, and in a very, very broken down schematic way, a Rubisco phylogeny looks a bit like this, where there's 3 typical forms or classes of Rubiscos that are typically distinguished from one another. And there is a single evolutionary interval, which I've highlighted here in red, that basically separates all Rubiscos that now contain a small subunit from those that do not yet interact with a small subunit. So coming back to the schematic I've shown you earlier, this would essentially be at the evolutionary interval over which the interaction, but also the dependence evolved. And for this, I have to also mention that basically all the Form I Rubiscos, so those that you see at the tips of these blue clades, all of those are already strictly dependent on the small subunit. Thus, both the interaction and the dependence evolved in this interval. The second question we then wanted to answer is whether or not we can create a non-obligate Rubisco heterocomplex. So something which we presume must have existed at some point in evolution, where basically Rubisco can interact with the small subunit, but does not yet strictly depend on it. And we presume that this must have existed at some point because it's pretty unlikely that both the interaction and the dependence on a novel interaction partner arise at exactly the same time. There likely was an evolutionary intermediate, even though in all extant forms of Rubisco, this is now lost. Third question was to then basically use this non-obligate intermediate that you see at the middle of this arrow here to then quantify the impact of the small subunit, because now we're basically, or we would be in a position where we can add the small subunit or leave it away and then assess the biochemical parameters of Rubisco. And then the last question we wanted to answer is why Rubisco started to depend on the small subunit. So basically following the arrow from the non-obligate intermediate to the dependence that you see all the way on the right. And now for the next slide, what we're going to do is we're going to zoom in into this phylogeny that you see on the top right of the slide, and basically zoom in on this red interval of evolution. So you're going to be seeing, or what you can see here is this last clade of Rubiscos that does not yet contain a small subunit. Then the evolutionary interval separating this from those that contain a small subunit, and then the two distinct clades of Rubiscos that contain a small subunit. Now, what we could do to basically answer all the questions that we had, or to try and find the substitutions that cause the interaction with the small subunit and also the dependence on it, is to just directly compare existing Rubiscos from these two clades with one another. The problem with this is that these sequences are much too divergent. They only share about 30% amino acid identity, which in Rubisco means that there's a total of about 300 amino acids that are different between proteins from these two clades. So it becomes really difficult to actually decipher which substitutions are causal, and you might be running into epistatic problems where you need several substitutions simultaneously to actually see the effects that you're looking for. The first thing I set out to do is I just basically looked in metagenomes to see if I can find novel Rubisco clades that weren't really characterized yet, that break down this evolutionary interval, so that are a bit more closely related to the first Rubiscos that contain a small subunit. Here I got fairly lucky, and I found 4 novel clades that weren't really characterized at the time that I found them, that all break this evolutionary interval and sit on the trajectory towards Rubiscos that contain a small subunit. Looking into the metagenomes of the organisms that encode these Rubiscos, we found that all the green clades down here don't yet encode for a small subunit, whereas this gray clade up here, which sits after the divergence into these canonical Form I Rubiscos that were known previously, they already contain a small subunit. And now with this information at hand, we basically got these genes, synthesized them chemically, and characterized the resulting proteins in the lab. And for this, we first set out to do in vitro stoichiometry measurements using mass photometry, just to assess when the oligomerization changed along this interval. Just for I know this is a refined seminar, but just for those of you that don't really know mass photometry yet, in a very, very, very broken down form, you can view a mass photometer like a nanodrop for proteins, that instead of measuring a protein concentration, it measures the mass of a protein complex. I can give you more details about the methodology later if you want, but I figured since this is a refined seminar, this might be good enough. Basically, with this nanodrop-like device, with the mass photometer, you're measuring a video that looks a bit like this, where each individual dot or each contrast point that you see right here corresponds to a certain protein mass or protein complex mass that you can then correlate back using a standard ladder or standard curve. And then the output is a histogram that looks a bit like this, where on the y-axis you have the number of events. This is just how many drops landed on the glass slide over which you're measuring. And on the x-axis, you have the inferred molecular mass coming from a standard ladder. And these types of histograms I will show a lot in the coming slides. However, not yet for these characterizations right here. These proteins we did all quantify by mass photometry, but to make a long story short, we found that this light green clade down here forms a simple dimer of large subunits. The clade further up actually forms the first clade of Rubiscos that is an octamer of large subunits, so 8 large subunits, just like in all the Form I Rubiscos. And then for this gray clade, even further up, we found that it forms this typical L8S8 complex composed of 8 large and 8 small subunits. Cool. So now that we've broken down this interval, what we could theoretically do, or is basically already propose an evolutionary scenario of how this canonical L8S8 Rubisco form evolved, in which a dimeric ancestral Rubisco just evolved for a bit and stayed dimeric based on the stoichiometry of its descendants. Then over this interval of evolution, turned into an octamer for the first time. And then in the next step of evolution, it likely took on its L8S8 form. So with this, we basically pinpoint the evolutionary interval over which the small subunit evolved to be between sort of this last green dot in the middle right here and this gray ancestor, which is the last common ancestor of all canonical Form I Rubiscos. Now, again, what we could do here is we could just take the extant sequences that sit in these clades and compare them to one another. However, there was still 2 divergence. So there were still about 220 amino acid differences between the most closely related proteins from these respective clades. And so what we decided to do instead is we decided to use a method called ancestral sequence reconstruction, with which we wanted to focus on ancestral proteins that basically don't exist at the node am I lagging? No, I'm not lagging, sorry. That don't exist at a leaf of this phylogeny anymore, but instead at an internal node. So basically, it's sad that I don't have a pointer here, but basically at these green and gray dots that you see in the middle of this phylogeny right here. The way this algorithm works is it takes all the sequence information in this tree, which comes from a multiple sequence alignment, which you feed into the algorithm to actually build such a tree. And then it takes the relationship of all the sequences, so how related a given sequence is to a given node within the tree, and then uses a probabilistic model of sequence evolution to back calculate the most likely sequence that would have existed at an internal node to basically explain the distribution of sequences that exist in present-day organisms with the highest likelihood. And so you calculate the sequences of these internal nodes, which represent ancestral proteins that would have, as such, most likely existed at some point in evolution in the past. All right, and so we decided to resurrect 2 large subunits here, this sort of green dot and then the gray dot that sits further up. And to go along with the gray dot, which we presume to have already interacted with a small subunit, we also resurrected the oldest inferable small subunit. For this, we did this entire procedure again, just with a different phylogeny, obviously built for the small subunit, but I'm sparing you all the details here. All right, and then we basically chemically synthesize these proteins from commercial vendors, or we buy them from commercial vendors as genes, produce them in E. coli, and then measure their stoichiometries. Our expectations going in is that UNC-L, so this is this green ancestor, which presumably existed before the small subunit first evolved, forms an octamer of large subunits, so 8 large subunits, which using mass photometry we could see is true. So we see we see a peak corresponding to an octamer and a smaller peak corresponding to a dimer. So that was true. And then we also expected that UNC-LS, ideally, so this is the gray ancestral protein, which we presumed already interacts with a small subunit, would form an L8S8 complex with the small subunit or the ancestral small subunit. And using mass photometry, we could also show that this is true and that it binds 8 small subunits and forms a stable complex in solution. Just to complement this, we also solved crystal structures of both proteins, and they do take on the typical stoichiometries that we would assume them to, or that we would hope for. All right. Now, there were 2 more outstanding questions which we wanted to answer, which is the first being, does UNC-L, basically this green ancestral protein which existed before the small subunit, can this by chance already interact with the small subunit? Here, once again, we used mass photometry, and what we did is we basically just pre-incubated UNC-L, so purified protein, with, in this case, a 4-fold molar excess of small subunit, and then looked whether or not we see a shift in mass on the mass photometer, which we didn't. So we concluded from this and other pull-down experiments that UNC-L cannot yet interact with the small subunit. And the second question was whether or not UNC-LS, so the gray protein further up on the tree, is already dependent on the small subunit. For this, I'm showing you gels right here, which basically just where I've loaded the crude and the soluble lysate. In the portion that you're seeing right here, this is just UNC-L, which doesn't interact with the small subunit yet. And you see that basically in the soluble lysate, you have proteins, whether or not the small subunit is co-produced. And then for UNC-LS, you see that there only is a visible large subunit band on the SDS page if you actually co-produce the small subunit. The same is true in native pages and Western blots, and also just in activity in clarified lysates. So on the right, you see that you only get activity for UNC-LS if you co-produce the small subunit. So from this, we concluded that ANCLS, the gray protein, is already dependent on the small subunit. In a nutshell, this means that we found the evolutionary interval over which both the interaction with, but also the dependence on the small subunit evolved, which was basically step one of what we wanted to find out. Step two was then to try and see if we can create a non-obligate Rubisco heterocomplex. This is basically the protein that we ideally want to study in the end. For this, we had a bit of a closer look at how separate these proteins are now from one another, so these ancestral Rubiscos that we're working with. And they're, in our case, separated by still a quite big amount of substitutions. So it's 95 substitutions and 3 insertions overall that separate these two proteins. But luckily, we had our crystal structures at hand, and we could first narrow basically our substitution set that we were interested in down to those substitutions that actually occur in proximity to the large subunit, small subunit interface. Because what we were trying to do now is we were trying to engineer an interaction surface into basically ANCL, into this green protein. So making it a tiny bit more like ANCLS, like the gray protein, but only at the apex of the complex by introducing substitutions that naturally occurred over this evolutionary interval. Looking at those 14 substitutions that localize in the large subunit, small subunit interface a bit further in the crystal structure, we could see that quite a few of these interactions quite a few of these substitutions don't actually result in amino acids that engage in any meaningful interaction between the large and the small subunit. So we took this, all of those substitutions that didn't really engage in any interactions, we took those out of the set of substitutions that we were interested in. This left us with 7 to 8 amino acids that actually engaged in a direct interaction between the large and the small subunit. And here I initially tried to reconstruct the interface in single or double substitution steps, which basically results in a plot like this, where I take ANCL, this green protein right here. I introduce all of these single substitutions that are listed on the left of this graph. One by one, I purify the protein and I test by mass photometry whether or not I see binding of the small subunit. For single substitution constructs, I couldn't see any binding. For double substitution constructs, I also couldn't see any binding by mass photometry. And then going into triple substitution constructs, I could first start to see some small subunit equivalents being bound to the protein complex, which gave us some hope. However, this entire procedure turned out to be quite tedious, and I decided to just introduce the 7 strongest binders, or the 7 amino acid substitutions that result in the introduction of an amino acid with the highest buried surface area at the same time, which resulted in a protein that we call ANCL plus 7. And ANCL plus 7 could actually bind 8 small subunit equivalents at very low concentrations, so at nanomolar concentrations. Looking at the mass photometry data of ANCL plus 7, if I purify it in the absence of a small subunit, it forms a nice octamer of large subunits. And then as soon as I add purified small subunit, this peak shifts. We get binding of 8 small subunit equivalents to the complex and the reconstitution of an L8S8 complex. Again, to further corroborate this data, we also solved crystal structures of the protein with and without the small subunit, and see that it basically behaves as we would expect it to, or as we would hope. So now with ANCL plus 7, we found a non-obligate Rubisco heterocomplex. And with this, we now set out to basically answer what the impact of interacting with the small subunit is. First obvious thing that we looked at was the structure. Since we had just solved the crystal structure, we had hoped that the binding of the small subunit would allosterically alter Rubisco in a way that would basically make it obvious for us to decipher a cause of the small subunit. However, what I'm plotting here is the active site of Rubisco, but over the entire protein, basically Rubisco doesn't change at all. Aligning the structure of Rubisco in the L8 or L8S8 form results in an alignment that has a root mean square deviation of less than 0.3 angstrom. So the structures are essentially identical, except for obviously an extra small subunit being bound in the L8S8 form. However, we still set out to see if there's any differences in catalysis, which would likely then stem from dynamic effects or effects that we don't see in the crystal structure. And here we did actually see differences. So what we're comparing here is the L8 to the L8S8 form, so with and without small subunit, of ANCL plus 7 in both cases. We see slight differences in the Michaelis-Menten kinetics of carboxylation, a slight decrease in the Km for CO2. It's roughly a threefold decrease in the Km for CO2, which comes along with a roughly 3 to 4-fold increase in the Km for RUBP. So this was already interesting to us. We assume that this makes it a slightly better carboxylase, and we can also see that it improves the catalytic efficiency overall, just by decreasing the Km for CO2, which probably was very important in environments where CO2 was limited for organisms. However, what we were more interested in at the time is to see whether or not the small subunit modulates Rubisco's specificity factor. I've mentioned earlier that Rubisco's small subunit was presumed to alter the specificity factor, but now we could actually test it. And when we first went into this experiment with L+7, we saw that there is a slight effect on the specificity, but it's not super dramatic. Specificity improves from roughly 23 to 29. So there's about a 20-25% increase in specificity. However, in downstream experiments, we found an additional substitution, which makes this L+7, +8, sorry, which actually further increases this effect drastically. And the addition of the small subunit then increases specificity from roughly 25 to about 40. Now, obviously, this probably doesn't mean very much to people that are not familiar with Rubisco, but this is roughly the change in specificity of going from the best Rubisco that doesn't yet interact with a small subunit to roughly the specificity of the worst Rubisco that interacts with a small subunit. So these are natural Rubiscos right here. So we can basically recapitulate the change in specificity that we would have expected to occur along this evolutionary interval with a few substitutions, basically 8 substitutions here to recreate the binding interface, and then the addition of the small subunit. Importantly, however, the small subunit alone is not the whole cause of increasing specificity, but there are certain substitutions on the large subunit that are required for this change in specificity that also importantly don't have an effect if the small subunit isn't present. You can see this here on the plot where basically you compare the condition where no small subunit is present between L+7 and L+8. Cool. One last thing that we asked is whether or not the small subunit has any effect on stability of Rubisco. And this was motivated by the fact that basically all of the Rubiscos that are closely related to the evolutionary interval that we care about stem from thermophilic organisms. However, when we just checked whether or not it changes the thermal stability of Rubisco, we saw that there's basically no difference whether or not we add the small subunit. Rubisco in this case is stable either way, but the small subunit doesn't improve stability. So what we've done here is we basically incubated Rubisco at different temperatures, then let it cool down, and then assayed the remaining relative activity. And here we don't see any difference. However, when we actually assay at different temperatures, so when we perform the assay of the catalysis assay at higher temperatures, we see that the small subunit actually massively stabilizes catalysis to higher temperatures, which allows catalysis up to maybe 80 degrees that wouldn't really be possible without the small subunit anymore. And this is obviously highly interesting to us because we think that the small subunit first evolved in thermophiles, and this would have immediately opened up new habitats to organisms that suddenly carried the small subunit and had the small subunit interact with this Rubisco. So as a conclusion, the small subunit was likely highly beneficial when it first arose, and it later enabled the adaptation to rising oxygen concentrations. All right, and now turning to the last question that we had, which was basically why Rubisco suddenly started to depend on the small subunit when it previously didn't. And here we followed or started out with a hypothesis which is called hydrophobic entrenchment, which basically states that anytime a protein, this can be any protein, this isn't specific to Rubisco, anytime a protein increases its complexity by either engaging in a new homo or heterooligomerization, in this case, what is depicted is a heterooligomerization, you create a new interface. And in this interface, you have a changed constraint on the protein or on the evolutionary sequence space that is available to the protein. In the absence of this interface, for example, you might not be able to accumulate hydrophobic amino acids on the surface just because they would make you aggregate. And then once this new interface is created, suddenly you are able to accumulate these hydrophobic amino acids just because they are now shielded from the environment through this new interaction. And this entire process can make protein-protein interactions become dependent or become obligate, just because now suddenly you can't take this novel subunit away anymore, just because the subunit that was previously able to function in isolation and soluble in isolation would now start to aggregate after it has accumulated hydrophobicity in the newly buried interface. So for this, applying this to Rubisco, we obviously hypothesized that this so-called change of constraint is likely largest in proximity to the small subunit. And so we focused on, this time, on all 14 interface substitutions that basically occurred along the evolutionary interval. So instead of building it up one by one, as I did earlier, we now decided to introduce all 14 substitutions simultaneously into ANC-L to create what we called ANC-L plus 14. And this protein, ANC-L plus 14, basically became fully dependent on the small subunit. So what you see right here on the left is ANC-L, just without these 14 substitutions, and there's no difference in solubility in lysates, whether or not the small subunit is co-produced. And then in ANC-L plus 14, the presence of the small subunit is essential. We then further tested whether or not single substitutions can be responsible for this by introducing all 14 substitutions individually. And here we found that a serine to tryptophan substitution, so this is exactly the scenario that we proposed earlier, basically a substitution that goes from a hydrophilic to a hydrophobic amino acid, causes a loss of about 75% of insolubility just with a single amino acid in a protein of 500 amino acids. And then if we combine these two substitutions that I've highlighted here, so this is E170N and the serine to tryptophan substitution, we actually abolish all solubility in two substitutions. And it's important to note that this loss of solubility is not because the protein is inherently misfolded or just unable to fold anymore, but it can always still be rescued by co-producing the small subunit. So what you're seeing here is a native page Western blot, where basically you see that if we co-produce the small subunit in this E170N serine to tryptophan mutant, we still get soluble Rubisco production. So it's not an inherently misfolded protein, it just depends on the small subunit for solubility now. And basically now we wanted to find out how this serine to tryptophan or how this double substitution construct actually makes Rubisco depend on the small subunit. And luckily in our case, the single substitution here retained roughly 20–25% solubility. So we could actually purify this and study the phenotype of it in vitro. And here, once again, mass photometry came to our rescue in a way, because as soon as I measured these proteins on mass photometry, I could see that there was an additional sort of oligomer observable that corresponded to roughly 2 times the mass of a single L8 complex. And just for reference, ANC-L without this serine to tryptophan substitution does not show this higher molecular mass complex at all. And then thirdly, I could also basically put this protein into 2 molar sodium chloride, for example, to push the population of higher order oligomers that we see. And as I've already sort of schematically alluded to here in these graphs, we assumed that what we're observing here is sort of this fibrillation-like assembly of large L8 complexes of Rubisco that would basically stack indefinitely. And we then looked at this using negative stain electron microscopy, which confirmed this fibr-like behavior. And then we're also lucky enough to solve a roughly 3 angstrom cryo-EM structure of this complex where it fibrillizes out. You can also see this here in an animated view, where basically we have just 2 repeats of this L8 complex stacking on top of each other to form this fiber interface. The fiber interface is driven by a cation-pi interaction, engaging into this interaction with the tryptophan 437 that we just introduced. So this also perfectly makes sense as to why we would now start forming these fibers. And importantly, if we try to model small subunits onto this fiber, the space between fibers is too small. So there's a big steric clash between interacting octamers, which basically prevents fiber formation as soon as the small subunit is bound. So this also explains why solubility is recovered if we co-produce the small subunit. Now, one last thing that we wanted to answer is whether or not the dependence-causing substitution, so for example, the serine to tryptophan, is beneficial in any way. So whether or not it's beneficial to the organism or for Rubisco in general to incorporate this tryptophan 437, which makes it depend on the small subunit. And here I'm only showing Michaelis-Menten kinetics without the small subunit present, but we basically see that there's no different kinetics at all. This is actually an overlay of the two different graphs, which look pretty much identical. So we couldn't really detect any advantages of incorporating the serine to tryptophan substitution, even though it makes Rubisco depend on the small subunit. So what we conclude from this is that basically over the course of evolution, you can neutrally accumulate these substitutions that cause you to depend on a novel subunit, just because as long as you've created this interface, they don't really incur a negative penalty on the protein itself. So they don't make you any worse adapted than you were before. The only thing that it causes is that you now suddenly strictly depend on your interaction to a novel interaction partner. And then very last thing we wanted to answer is whether or not more of these sort of tolerated neutral substitutions accumulate. And what we set out to do here is we basically just looked at our sort of evolutionary interval again. And so far, everything that we've done was essentially taking Anc-L and introducing substitutions that occur along the interval into it. And now instead of going in that direction, what we decided to do instead is we decided to take Anc-LS and then take out substitutions to try to recover solubility without the small subunit. Here initially, we looked to take out two substitutions. Those were the two substitutions that made the Anc-L+7 depend on the small subunit. But then in the next step, we also decided to take out a whole set of substitutions, which basically were the substitutions that occurred in the fiber interface, which we've identified in the cryo-EM structure. And this turned out to also be 14 substitutions coincidentally that are, we've also decided to take out these 14 substitutions that are in the fiber interface. And now this is basically an SDS page, so showing the solubility of large subunit Rubisco in soluble lysates when the small subunit is not co-produced. And you see there's basically no large subunit band for Anc-LS alone. If you revert either single substitutions or the double substitution constructs, you gain some solubility, but you don't get all the way there. And in order to regain full solubility, you would actually need to revert basically all, well, in our case, we reverted all 14 substitutions in the fiber interface, but you definitely need to revert more than the two substitutions that it took to basically first make you depend on the small subunit. So solubility is only fully recovered when reverting more than two substitutions. All right, and then one last thing is that we basically realized that this fiber interface that we were looking at doesn't actually overlap with the large subunit, small subunit interface so much. So even though we reverted 14 substitutions, the large subunit, small subunit interface is still in place. And this fiber interface reversion construct can still actually bind the small subunit. Looking at mass photometry of this, again, the fiber interface reversion construct forms a happy octamer in solution. And when we add, in this case, a 10-fold molar excess of small subunit, we can get this very typical shift and reconstitute the L8S8 complex. So far, this was not unusual. This was as expected. However, what was interesting is that in this case, now this protein, this fiber interface reversion, strictly depended on the small subunit for catalysis. So basically, you see right here, the protein, as soon as we add the substrate, ribulose 1,5-bisphosphate, we would typically expect to see turnover or production of 3-phosphoglycerate. And this didn't happen until we added the small subunit. And this is unlike anything we had seen before, just because in the past, we had characterized Rubiscos without the small subunit and then added the small subunit, and catalysis didn't really change so much. What this meant to us is that actually over these 81 substitutions that have occurred between Anc-L and this fiber interface reversion construct that we've just sort of created and talked about, a further set of substitutions must have arisen or accumulated that also now make catalysis depend on the small subunit, as opposed to only solubility. So the conclusion from this is that I think it's interesting that basically as soon as you alleviate a certain constraint, you immediately start accumulating substitutions that erode, for example, solubility or even the ability of a protein to catalyze its reaction on its own, just by accumulating these substitutions, because you can. Now, as a conclusion to all of this, Rubisco's small subunit improved catalysis as soon as it arose at physiologically relevant conditions, mostly by decreasing the Km for CO2. It enabled the evolution of higher specificity. We think that the small subunit became essential mostly due to conditionally tolerated substitutions, so those that don't really bring a positive effect, but just are tolerated because they don't incur a negative effect. And in general, these accessory subunits, these novel binders, can be important sources of functional novelty, as we've seen that specificity, for example, probably cannot evolve in the absence of the small subunit. Now, as a small fun addendum, just because I didn't really get to talk about the exact timing of all of this, I want to quickly mention when all of this happened. So going back to this phylogeny that I've been showing all over the slides, we know from previous publications that the diversification of Form I AB Rubiscos, so those are those Rubiscos right here, roughly coincides with the Great Oxygenation event. So the point where the orange arrow is pointing to right now is roughly the timing when people think the Great Oxygenation event first happened, which was about 2 to 2.5 billion years ago. And as you can just basically see on our phylogeny, the Rubiscos that we investigated are older than this, so they presumably evolved and existed before the Great Oxygenation event, if these inferences from previous publications are true. This basically means that Rubisco started to improve its specificity, so its ability to discriminate between CO2 and O2, before oxygen was really present in very high concentrations or in problematic concentrations. This could have two implications, there might be two reasons for this. The first one would be that there was early localized oxygenation, and Rubisco just happened to first evolve its small subunit in an environment where oxygen was present by chance, before the Great Oxygenation event, so before the atmosphere became oxygenated. Or this might be a form of pre-adaptation, where Rubisco essentially recruited the small subunit for its effect on stability of catalysis at higher temperatures that I've gone into in the results, and only then later started to use the small subunit for its effect on specificity. Yeah. All right, and with this, I'd like to come to the end. Thank all my amazing lab members and mentors, most importantly, Georg Hochberg and Toby Erb, and then also Jan Zasicki, Thomas Heimel, Simone Prinz, Sijun, and Oliver Müller-Kayar, who've helped massively with all the data curation measurements and just have assisted me very much. Thanks for listening.
Thank you, Luca, for your informative presentation. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for today. Let's get started. Our first question is, do you think or know whether SSU also underwent similar progressive mutations accompanying the changes in Rubisco?
Right. I don't know, but I definitely think it did. It's shown in like much higher up the phylogeny of Rubisco in plants, for example, that specific substitutions in the small subunit still have an effect on catalysis. And even just replacing small subunit homologues that look very similar to one another also affect kinetics of Rubisco. So there certainly was selection on the small subunit as well. And I'm sure that the small subunit also co-evolved with the large subunit to a certain extent. It just, in this study, we couldn't really deal with that because it just increased the combinatorial space too much. But this is certainly an interesting question that should be followed up on, yes.
Great, thank you. Our next question asks, are the mechanisms of how protein-protein interactions become essential, generalizable, or confined to Rubisco?
No, I definitely think they are generalizable. Basically, if I basically this mechanism of hydrophobic entrenchment that I've showed earlier, this should be completely generalizable to any sort of protein-protein interaction, protein-protein interface, just because it's something as universal as changing amino acids towards hydrophobicity. And since all organisms use the same, or roughly the same code of genetic code and roughly the same amino acids, but basically the same 20 amino acids, this should be generalizable, yes.
Great, thank you. What was the role of mass photometry in determining which substitution to focus on or investigate further?
Yeah, well, basically, I mean, I think I showed this in the talk, but we massively relied on mass photometry for basically assessing the phenotype of our substitution constructs, and also for finding basically also the cause of the dependence onto the small subunit. So mass photometry was kind of essential, and every time we did any substitution, the first thing we did is to just throw it on the mass photometer, also because it's so quick and easy to just measure something. So it sort of just became a go-to practice to just measure everything by mass photometry before we actually move on with doing any biochemical experiments with it. And sometimes this gave us really fascinating insights, such as, for example, this polymerization that we could have only found by mass photometry.
Great, and it looks like we have one more question here today, and it asks, is the approach of ancestral sequence reconstruction universally applicable? What are determinants for its success?
Yeah, so theoretically, ancestral sequence reconstruction should be universally applicable. The problem is that you need a well-sampled protein family, ideally, just because the more sequences you have available, the easier it is to actually then build a tree that has enough granularity to actually find nicely defined evolutionary intervals that you want to investigate. And the more information you have available in terms of sequences, the more robust your reconstructions actually become. So all of this plays into how successful a ancestral sequence reconstruction venture is going to be. But in theory, it should be universally applicable. However, another component to all of this is that fast evolving proteins might be a bit more difficult to reconstruct, just because along a certain evolutionary interval, more substitutions are likely to occur. In Rubisco, this is actually great because Rubisco is a rather slow evolving enzyme. So we don't really expect to see so many substitutions along a certain evolutionary interval, even though we were still dealing with 95 differences between the two proteins that we cared about.
Great, thank you. It looks like that wraps up our Q&A session for today. Luca, do you have any final comments for our audience?
Thanks for listening. Not really, no.
Great. Thank you again, Luca, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Refine, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.